As much as I would love to continue the politics talk. It's very important. I'm not making fun. I'm serious. All right, guys, let's get started. We are really switching gears here now and finally done with neuro. I know you'll be happy with that. And we're going to discuss something completely on the other end of the spectrum, the blood system, hematologic system, so structure and function of that. As always, we're sort of, going to do our first lecture today of sort of laying the groundwork. And then as we move forward throughout the week and get together back on Thursday, we'll talk more about alterations of the hematologic function. So you have 6 objectives here regarding Chapter 19. And when we really start to sort of, hone in now and start to really think about the functions and sort of the purpose of the hematologic system as we move forward today, what are some things that really come to your mind in terms of sort of the key functions, the key roles of our blood? And, and what I mean by that is any of the constituents, the plasma, any of the blood cells, etc. What do we really want to be working properly so that as we start to talk about pathophysiology regarding the blood of things that could go wrong. So let's talk about things that go right first. What does our blood do for us? Carries oxygen, specifically on what or with what? The hemoglobin on the red blood cells. Good. That's a key one. What else? Right, nutrients to the tissue, and then toxic sort of waste products and carbon dioxide and things like that away from the tissue to be expelled and gotten rid of throughout the body. Good. What else? Immune functions. Why? Right. Right, all the white blood cells, leukocytes, lymphocytes. Good. Good. Clotting factors. That's a big deal. Right. Very good. What else? Those are the big ones. There's a few other ones that may not be quite as straightforward or quite as, first to think of. Anything else you can think of? Yeah? Good, good. There was talk about it sort of being like a carrier of some of the waste material out, but it also can filter. It also filters. Good. Or the blood, and also I should say, any of the old blood cells, so, broken down red blood cells, broken down platelets, things like that can also get filtered out. Good. And then one more that maybe isn't as obvious. Good. Very good. Acid-base balance. Very good. Our buffering systems, right, that we talked about last semester when we talked about acid-base and electrolyte balances, those are a key sort of function of our blood hematologic system as well. Excellent. Very, very good. All right. So that being said, here's some of them, but you guys have listed more, so that's great. That's very, very good. All right. So when talking about sort of the composition of blood, really, and, this isn't any big surprise, but blood is basically, 90% water, right? With just a small little other percentage of being the solutes floating in that liquid, in that plasma. So we're talking about, like, at any given time in the human body, in the adult human body, we're talking about blood being about 5.5 liters. So that's just to kind of give you a reference when you start to think about blood loss or blood donation or hemorrhage or whatever, about how much blood we're really talking about in terms of, sort of compensating for whatever blood loss may have occurred. Plasma is the big sort of, water component of the blood. 50 to 55% of the blood volume is plasma. And as you know, it's filled with sort of organic and inorganic elements that sort of help us throughout some of these processes that we're talking about today. So within the plasma, then you have some of those specific elements. So one of the biggest things that you're going to find in the plasma are plasma proteins, right? And I want you to be familiar with these because these really do play a key role into some of these major functions that we're going to be talking about today. So first one, albumin, right? I hope at this point I have slammed you over the head with albumin at this point. I hope I don't have to really discuss what that does. But again, has to be mentioned when you're talking about plasma proteins. Another really big component, or another big type, I should say, of plasma proteins are globulins. These are, most of the time, these are sort of like our carrier proteins in the blood. And the biggest, obviously most sort of known example of globulins in the plasma are our antibodies, right? Are our immune globulins. So those are certainly a big component of the plasma and, as a plasma protein. And then lastly, clotting factors. So mainly fibrinogen. Fibrinogen is what promotes coagulation and stops bleeding from damaged blood vessels, right? So that's also considered a plasma protein. And we'll talk more about clotting factors and fibrinogen later on when we talk more about hemostasis and the clotting process. But good. So plasma proteins aren't necessarily just your albumin, and that's what I wanted to be clear about today as we start to talk about other things that you can find in the blood that certainly help with some of these processes that we're talking about today. Good. Okay, so there's your plasma proteins in a nutshell. And then we start to talk about other components of the blood and start to talk about our actual blood cells, right, the cellular components. So when I am talking about that, I mean red blood cells, white blood cells, platelets, okay? So here we're going to start talking about first the erythrocytes, the red blood cells. Now, for each one of these cellular components, red, white, and platelets, I want you to make sure that you are intimately familiar with the ranges, right, of what we really want to see in terms of, normal lab values. You will be responsible and sort of, held accountable for those on the exam, and then, also more importantly for the rest of your life, right? Not so much for the exam, but I'm sure your patients would be appreciative if you understand what the normal lab values for these cells are. So, red blood cells, certainly most abundant cell in the body. They are responsible, as we have already discussed, for tissue oxygenation, specifically because of what's found on red blood cells? Right, very good. So I want you to sort of understand that. So it's one thing to just kind of say, yeah, we know that hemoglobin's on the red blood cell, and that's what we're concerned about in terms of oxygen carrying capacity of the blood. But how much oxygen our body tissues get does very specifically depend on how many red blood cells we have and if they're working, right? And not only just how many red blood cells and if they're working, but how much hemoglobin is on those red blood cells, okay? So, and that's obviously what dictates how much oxygen can get to the tissue. So what I really want you to think about is the relationship between, yes, red blood cells, but also how much hemoglobin is on those red blood cells, because you can have a lot of red blood cells and not have adequate hemoglobin content, right? Or you can have perfectly fine hemoglobin content, but unfortunately not enough red blood cells circulating in the blood, right? So we need to talk about the relationship between adequate number of red blood cells, which also has adequate number of hemoglobin molecules, which also then has adequate oxygen sitting on that hemoglobin to be able to really get the oxygen to the tissue. And so we'll talk more about that when we talk about the different types of anemia on Thursday, and that you can have some that are sort of normal red blood cell numbers, but the hemoglobin on it is crappy, and vice versa. So I just wanted to make sure we're clear about that as we move forward. That responsibility for tissue oxygenation is not just quite as straightforward as you may think. Also, the shape of red blood cells are really important. So what do I mean by biconcavity? What do I mean by that? Why is that shape? I think we sort of know the shape of a red blood cell, right? Kind of flat and round, almost like a donut that has a little bit of a, concave in the middle, but it's not completely a hole. Okay, we know the shape, but why is that shape so important? Someone say it clearly so I can. Right, exactly. So not only can that biconcavity, the size and the shape, ideally be suited to sort of function as a gas carrier and be able to sort of increase its surface area and its ratio to be able to hold more gas and different types of gas on that shape. But also, it's also responsible for reversible deformity, which is exactly what was just mentioned. That it's able to sort of squeeze and almost become almost like a little torpedo, right? It can sort of squeeze down into these sort of funky shapes to squeeze through the small little bits of circulation, right? Into those little microcirculation, into those capillaries, and really get into the tissues and deliver that oxygen. And then obviously take the waste product out as well. So not only the shape, but its ability to have sort of what we call reversible deformity is what makes it an excellent gas carrier, and what also makes it excellent in terms of getting into small little nooks and crannies and spaces in the microcirculation and actually delivering that gas. So very good, very, very good. And you may have already known this as well too, but red blood cells actually have a pretty short life cycle as well, so about 120-day life cycle. And that truly is because red blood cells cannot really undergo mitotic division. Therefore, they have a pretty limited lifespan. And that's important to sort of keep in mind and be thinking about as well. Again, next class period on Thursday, when we start to talk about different reasons for anemia and different reasons for why someone might have a lack of a number of red blood cells, this short lifespan certainly doesn't help that, right? When we're talking about red blood cells getting destroyed for whatever reason, the fact that it can't quickly sort of have a large number present at any given time can be a problem. Good. All right. Any questions about sort of the sort of basic erythrocyte information? Good. A lot of this is review. Good. Okay. Moving on to leukocytes, your white blood cells. Here's your sort of normal white blood cell concentration to be responsible for. We know, right, this is just, almost like high school science sort of review, that the white blood cells, the leukocytes, are what defend the body against infection and remove also debris from those cells, or from the body, I mean. Okay, when thinking about white blood cells, when thinking about leukocytes, you really can classify them according to two different kind of categories or two different umbrellas. The one category is that you can classify the different types of white blood cells by their structure. And what I mean by that is either granulocytes or agranulocytes. Or you can classify them according to their functions. Oftentimes you'll see white blood cells sort of classified that way as well. And what do I mean by function? What do you think is a way that you could sort of extrapolate out two different types of white blood cells? Anyone know? Yeah. Sort of when you're thinking about like the different types of immune system. Yeah, that's true. Adaptive or immune. Mm-hmm. Right. That's what I was thinking about. Even more specifically, maybe even more on a basic level, the fact that are they phagocytes or are they immunocytes? Right. Good. So you guys are both kind of saying the same thing. Very good. Okay, so either structure, are they granulocytes or agranulocytes, or their function, are they phagocytes or are they immunocytes? Good. Very good. All right. So the way I'm going to kind of go through them this morning is talking more about the sort of granulocytes versus agranulocytes, okay? So granulocytes are those white blood cells that are membrane, they have their sort of membranes that are sort of hold those little sacks of goodies. Remember I talked about this when we talked about inflammation last semester, right? Granulocytes have membrane-bound granules in their cytoplasm within the cells themselves. These granules that are held in these little baggies inside the cells contain enzymes capable of destroying microorganisms. So they sort of release the bag, and sort of the bag breaks open, and those enzymes go to whatever microorganism is present and attack, right? So because of that, granulocytes obviously have inflammatory and immune functions to them. And they're also capable, and this is a good, sort of a very good function of granulocytes, is they're capable of something called amoeboid movement or diapedesis. Anyone know what that is? In terms of how granulocytes move around in our system? Anyone know what I'm talking about? Okay, this type of movement, this amoeboid movement, is basically stating that leukocytes are able to navigate through the vessel walls and right into the sites where they need to go, right into the interstitial space wherever the infection or the microorganisms are. So they're basically able to just kind of walk right through the vessel wall and out into the interstitial space or out into the tissues. So the way that I sort of think about it is they're very sort of Patrick Swayze and Ghost, right? Can just like walk right through a door, walk right through a wall to get wherever they need to go to sort of release those granules and start fighting that infection. So that's kind of what amoeboid movement is, and that is a classic sort of function or characteristic of granulocytes. Okay, so definitely, looking at the immuno aspects of things is certainly a big player that granulocytes definitely are. Okay, so the types of granulocytes, right? On this screen we talk about neutrophils and eosinophils, and again, we're not going to spend a lot of time on this. We talked about this a lot when we talked about immunity last semester, but neutrophils obviously in some sort of infection are the most numerous and the best understood of the granulocytes when talking about some sort of microorganism invasion. They're part of something called, or they are, shall I say, a polymorphonuclear neutrophil. And we're going to talk about that in just a little bit. And they are certainly those big phagocytes in early inflammation. So they're very helpful in an early inflammatory sort of response. Very soon, after specifically like a bacterial infection or invasion into the tissue, neutrophils migrate out because of that amoeboid movement, right? Straight out of the vessel, migrate out into that damaged tissue where they ingest and they destroy and they contaminate any of those microorganisms and debris that might be out in that tissue causing problems. So that's what I want you to really think about in terms of the function of neutrophils. So again, probably the most numerous, best understood of the granulocytes, and a big heavy hitter specifically in bacterial infections. Okay, any questions about neutrophils? Yeah. Just what I just said? Yes. Best understood, most numerous, right before that?
Like the digestive.
Oh, okay. Yes. Gosh, I have to remember what I just said. Okay, so they migrate out of the capillaries, they migrate out of the vessels into the tissue, wherever that microorganism may be, and they ingest, because they are phagocytes, so they ingest, destroy, and sort of contaminate, or sort of kind of start to break down those microorganisms and debris that are actually causing the problem out in the tissue. Does that help? Is that what it was? Okay, good. Don't even know the words that are coming out of my own mouth sometimes. Okay, so that's neutrophils in a nutshell, right? Again, to be clear, big heavy hitter in bacterial infestations, if you will. Okay, now how about eosinophils? Okay, so we also talked a lot about eosinophils last semester. And we see these a lot. We see the eosinophil count being quite elevated in an immune globulin E hypersensitivity specifically. Anyone remember what might be sort of an example of an immune globulin E hypersensitivity? So when clinically may you see eosinophils be quite elevated in, lab values? Yeah. Parasite infection, very, very good. Yep. And there's something else that might even be more kind of routine or normal than that. Allergic reactions, good. And even more specific than that, oftentimes there is some sort of allergic component to like an asthma attack, right? Because of the allergens that may irritate the airway. So you often see eosinophils quite elevated in asthma attacks as well, because they are kind of an example, if you will, of maybe some type of allergic reaction to something in the environment. Again, so eosinophils are elevated in parasitic invasions, allergies, and specifically, even more specific than that, asthma. Okay? Very good. So very relevant clinical example of that is, check if someone's coming into the emergency room with an active sort of, allergic reaction, or someone's coming in with an active asthma attack. You would draw their blood and look at do a CBC with differential to see all the different types of white blood cells that are present, and you would certainly see an elevated eosinophil count. So that's very relevant clinically speaking. Good, okay. Other examples of granulocytes are mast cells and basophils. Okay, so again, I hope I don't have to spend a lot of time talking about mast cells today, because we talked about it a lot last semester in terms of inflammatory. So you tell me really quickly, when might you see mast cells? First, answer that question for me. When are we going to see a high number of mast cells? Allergic reactions and just sort of inflammation in general, right? And what is then clinically, what is the manifestation of sort of hyperactivity of mast cells? Say that again. The signs of inflammation, right? Because mast cells release what? So they're granulocytes, right? And what do they release out of their little baggies of good? Histamine. So you're going to see all those responses of the body to histamine, right? So vasodilation, leaking of fluid out of the capillaries into the interstitial space, swelling, etc. Right. And that's what you're seeing with the activation of mast cells sort of being broken open, or shall I say, breaking open the histamine from the mast cells. Good. And basophils actually have structurally and functionally have a lot of the same similarities to mast cells as well. So if you think of sort of the functions of mast cells, you kind of understand the functions of basophils as well. Good. Very good. All right. So So are you starting to see here now, even as we just sort of end our discussion regarding granulocytes, the way that a lot of the things that we have talked about already this year sort of come full circle, they sort of play into each other. You know, talking about immune functions last semester to now blood functions of the blood this semester, seeing the relationship clinically of how these things sort of present and manifest in our patients. We're really kind of starting to put the pieces together now. And as we continue to move even more forward throughout the semester and talk about other systems, be it renal, be it bone, be it, you know, hormonal endocrine stuff, you're really going to see even more so things really kind of starting to come together, really putting the pieces together, that all of these things kind of work in harmony, or in pathophysiology, not so much in harmony, because of the way they all work together. So I hope that you're kind of just starting to see that big picture now. Good. I think you already are. All right. And then some examples of agranulocytes, right, would be specifically to talk first about monocytes and macrophages. Okay, so let's talk a little bit about that first. So monocytes are basically your immature macrophages, right? So they're formed and released by the bone marrow into the bloodstream. And as they mature, monocytes migrate into a variety of different types of lymphoid tissue. So you're going to find a lot of these in the process of maturing monocytes in places like the liver, the spleen, and in lymph nodes throughout our body, in various lymph node places. And there, in those lymph organs, is where they fully mature into tissue macrophages, okay? So again, the way I want you to think of it is monocytes are very immature macrophages. Monocytes are found in the blood because they're in the blood migrating to these lymphoid organs. Once they reach those lymphoid organs, they can sort of continue and finish their process of maturation, and they become macrophages. And it's really macrophages' role to sort of fight infections and be sort of functional in the tissue. So monocytes are in the blood, macrophages are in the tissues in the body. Did you have a question, Taylor? No, okay. Thought I saw a hand out of the corner of my eye. Okay. Yeah. Well, now that you mention it.
Talk about like when we pull these labs, what they mean, or is this?
We're going to talk about that on Thursday. We're going to talk about seeing like if you have increased monocyte counts in your blood. Yeah, we're going to talk about that on Thursday. We're going to lay the groundwork today and start talking more about that on Thursday. Yes. And if I still don't answer your question on Thursday, ask it again. Okay. Now that you mention it. No, it's fine. I'm just teasing you. Okay. So her question was, are we going to sort of talk about how if we see different counts or differentiation of increased monocyte count versus an increased macrophage count in the blood, what does that sort of mean? We're going to talk about that on Thursday, and if I still don't answer your question, let me know. Okay. Okay, so that's your monocytes and your macrophages. Lymphocytes are those immune cells that we talked about last semester, right? So your T cells, your B cells, your plasma cells. And really, and we talked about this last semester, but really most lymphocytes circulate in the blood. They eventually come to reside, again, also in some sort of lymphoid tissue, so liver, spleen, lymph nodes, etc. And so they reside in the lymphoid tissue as they mature into T cells, B cells, and plasma cells, okay? And we're not going to really spend a lot of time talking about natural killer cells today, but I will tell you they oftentimes will sort of resemble lymphocytes, and they are sort of kill certain types of tumor cells. So they very oftentimes in our immune system will run sort of like surveillance to make sure that there's no sort of abnormal cell growth and no sort of abnormal tumor growth occurring. And they actually kind of stop that growth and block that growth from occurring. So they're actually, like I said, sort of work as sort of security guards in our system, in our immune system, to sort of kill any abnormal cells that may be growing. Okay, and those are your, again, top 4, if you will, agranulocytes. Now, also when discussing agranulocytes, I want to talk a little bit about this mononuclear phagocyte system, which I kind of mentioned a couple of slides ago. So, oftentimes you'll hear the mononuclear phagocyte system referred to as something called MPS. And what it basically is that it consists of a line of immune cells that originate in the bone marrow, that are then transported into the bloodstream and differentiate into monocytes, as we just sort of mentioned, and settle in those lymphoid tissues as mature macrophages. Okay, so I just said that. But what then do they do? What is the sort of role of some of these cells that we've already talked about? So cells of the mononuclear phagocyte system are basically our filtration of our blood system. They are very, very responsible for filtering blood. So cells of the MPS ingest and destroy microorganisms and any foreign material that may be found in our blood. Okay? Again, most of these cells, so the MPS, the system is mostly in the liver and the spleen. And again, what I want you to understand about this is that the mononuclear phagocyte system filters and cleanses the blood, not only of, as I have mentioned here on the screen, microorganisms and foreign material, but it's also the system that takes out of circulation and filters out of circulation our own dead blood cells. So those broken down red blood cells that I just said have a kind of short lifespan, they take out those old broken down, not functioning, old red blood cells. Same thing with broken down, non-functioning platelets. They'll filter out, especially in the spleen, this system will filter out some of our own debris, our own cells that are broken down and not functional anymore. So that is what I want you to understand about the mononuclear phagocyte system. It certainly does fall under the umbrella and fall under the function of some of these agranulocyte cells. Yeah? Yes. No, not necessarily. They're a little bit different. The Kupffer cells are specifically in the liver, like we talked about, and sort of work as sort of that, sort of immune aspect of the liver. But these are not necessarily just in the liver. In fact, most of these you're going to find in more of the lymphoid tissue, like just the lymph cells, and also specifically the spleen. And we'll talk about that in just a second when we talk more about lymph nodes and spleen. Mm-hmm. They all work together in this system to do this filtration of debris and dead old cells. Yeah. Mm-hmm. Good question. All right. So again, I just wanted to sort of quickly mention that before we move on to talking about a different type of cell in the blood. I wanted to be clear that this sort of falls under the agranulocytes, under the white blood cells. Okay. So again, mostly this process is done in the liver and the spleen. And hopefully that should make sense to you because that's where these mature macrophages are, right? We just talked about that. And the macrophages are one of the big sort of heavy hitters in terms of being responsible for this filtration system. Okay? All right. Then, if that's clear to you, hopefully, we'll talk about our last component of sort of the cellular component of the blood, and that's platelets, okay? So here is your normal lab range for you. Now, interestingly, platelets are not true cells. They are actually cellular fragments. And we'll talk a little bit about why that is in just a second. So they are disc-shaped sort of cytoplasmic fragments. And we know, obviously, that these are the cellular components of the blood that are essential for coagulation and control of bleeding. They are very much caused, or shall I say, controlled by thrombopoietin. Thrombopoietin is a hormone growth factor, so it's part of almost like the endocrine system. It's actually like a hormone that controls blood clotting, that induces platelet production in the bone marrow. So controlled by thrombopoietin, and thrombopoietin regulates the platelets by inducing platelet production in the bone marrow. Okay? Do I need to say that again, or are we good? Say it one more time. Okay, so thrombopoietin is a hormone that is sort of the main regulator of platelet formation or platelet production. And the way that it does that is by inducing platelet production and activating platelet production in the bone marrow. Okay. And to kind of go back to why they're actually not true cells and they're these sort of cellular fragments, is that platelets they attempt to go through mitotic division. So the DNA, the genetic information actually does kind of double and sort of pull apart inside the cell the way normal division would occur. But instead of adequately then doubling and sort of pulling apart into good, nice new cells, they sort of break apart and fragment irregularly. And they're these cellular fragments instead of true functional cells as we think of them. Most of the platelets in our circulation, or in the circulatory system, I should say, are actually out circulating at any given time. They're out floating around in the circulation. But what's interesting is that there is, besides the platelets that are out sort of circulating, there's an additional like 1/3 of that circulatory platelets that are available and sort of in this reserve pool sitting in the spleen. So 1/3 of the platelets available are just sitting in the spleen, in this sort of reservoir, in this pool. And we'll come back to why that may be important in just a little bit. But for the other platelets that are out there, they circulate for about 10 days-ish. Then they get old, or they start to be, dysfunctional and no good anymore. And then they also get filtered out by the MPS, by that system that we just talked about in the spleen. So important to understand that as well. Okay. So, given that information regarding all of these sort of cellular components of the blood, I want to kind of stop there for a minute and move and talk about some of these lymphoid organs that play a big function in our circulatory system, in our hematologic system. So we're moving away from the actual components of the blood for a minute, and we're going to talk about some actual organs that also help in this hematologic process. So the first organ that we're going to talk about is the spleen, by far the largest of the lymphoid organs, right? Now, interestingly, in terms of its functions, it is the site of fetal hematopoiesis. Now, what do I mean by hematopoiesis, first of all? Creating red blood cells, or just really blood, in this case, blood in general, right? How is that, so it's the site of fetal hematopoiesis. How is that different than once the baby is born, the umbilical cord is cut, then where does all of our hematopoiesis occur? Bone marrow. Very good. Okay. So the spleen works as the site of fetal hematopoiesis. It also is that functional role of the MPS, filtering and cleansing the blood. And its lymphocytes mount immune responses to any bloodborne microorganisms that we sort of, come into contact with, and serves as a blood reservoir. So, we've just now said that there is a big component of platelets sitting in the spleen. I've also now just said that there is a big sort of reservoir of blood in general sitting in the spleen. Why are we so concerned then about any sort of injury to the spleen? What do you think? Yeah. Right, exactly. So let's say and I have often seen this happen in terms of, traumas and things like that. If you have some type of splenic lac or ruptured spleen or something like that because of a trauma, why are we concerned? Well, one, because we're at an increased risk of a lot of blood loss and hemorrhaging now because of this big pool of blood was sitting in there. This big reservoir of blood was sitting in there that is now dispersed, right? We're also concerned about issues with clotting now because there was a big, good old reservoir of platelets in there that have now been destroyed or released through this splenic injury. And then also big, houser here of lymphocytes that are able to sort of mount immune responses to whatever our body may come into contact with. So now we're talking about lack of immunity, right? Major hemorrhage risk, major issues with clotting, and major issues with immunity now when you've got a major injury to the spleen for all of those reasons. Very good. Good. Okay, so those are kind of the major roles of the spleen. Therefore, extrapolate that one step further and start to be concerned about why we're worried about injured spleens, pathophysiologically speaking. Good. Okay, let's talk more about the sort of structure of the spleen, so to speak, and how hopefully some of these roles will become clear to you when you talk about how it's actually sort of, organized, so to speak. So, the spleen is a huge lymphoid organ, as I mentioned, and it's a very big filtration organ because of something structurally called splenic pulp. What splenic pulp basically are these, this sounds lovely, doesn't it? Delicious. It's these masses of lymphoid tissue that contain those macrophages, right? That filtering, those macrophages that we talked about, and lymphoid tissue. And what happens is, as blood enters the spleen, it basically circulates through this pulp. And I honestly think about it, sort of the pulp acting as like a screen like literally like the screen on your window kind of thing. As the blood sort of circulates through that pulp, through that spleen, or through that screen, it certainly is one of the chief sites of filtration, of immune and phagocytic function, because you're literally filtering this blood as it gets pumped through this screen, so to speak. So that's kind of one of the biggest sort of functions of the spleen. So again, if you think about some sort of damage or laceration or rupture of the spleen, you're losing all of that good pulp and all of that filtration process, okay? And then the other thing that, and I have a picture of this in just a second, so some of this is going to come alive a little bit more. The other big structural aspect of the spleen is that it has what we call venous sinuses. They're literally like little cavities, like little sinuses inside. I'll go to the picture here. Inside the spleen. This is actually a picture of a lymph node, but it's the same makeup essentially as the spleen. So see how you have these, like, this is sort of the pulp, right, in here, that blood gets filtered through and sort of screened through. And then you have these sinuses. You have like these little cavities. And this is where phagocytosis of those old, damaged, icky, non-functional blood cells come to sort of be filtered out and destroyed. It also is the area where you have that blood storage. So this is where you have that reservoir of blood sitting. And again, this is a picture of a lymph node, so I don't want to confuse you, but the structure is very similar to that of the spleen as well. And in these sinuses, in the spleen, you can actually hold up to like 300 mils of blood. So again, think about if you have some sort of damage to the spleen, how much blood loss you're losing right there, right? How much blood you're at risk of hemorrhaging out. Okay. So again, the spleen is divided into compartments that contain masses of lymphoid tissue, and blood-borne antigens encounter lymphocytes here and initiate that immune response. So that's why the spleen is sort of considered such a sort of immune organ in and of itself, is because that's where you find a big body of lymphocytes there. Yeah. And so great. Oftentimes I've seen splenectomies because of pretty profound damage done to a spleen. So then what are we worried about on these people? All those things we talked about, right. And actually you bring up a good point. So I don't see it a lot, obviously, in the ER, but once these trauma patients, go to the OR, have their spleen removed, and now are in a trauma ICU or just on a trauma normal floor, you'll see in their orders, these are, adults that have had all of their vaccinations as a child, all of that has sort of been done in their life. And you'll see in their orders now this huge list of all of these weird vaccinations that they're now supposed to get. And a lot of that is because of their I shouldn't say complete, but a pretty profound lack of immunity to a lot of different things. And so you'll see these adults getting these really weird vaccinations that they normally wouldn't need or wouldn't need to get because of the lack of a spleen. So we really are worried about a lot of immunity issues with someone who does not have a spleen. So yes, you can live without one, but yes, you're going to have all of these complications that we've kind of talked about. Yeah, good question. Yeah.
This is definitely a stupid question.
No.
When your spleen, you're saying that you have an injury to your spleen,
Yes.
and then you're at risk for hemorrhaging. Like, I don't understand where that blood is going.
That's
And why is
coming
the problem?
from the spleen, you mean?
Is it just busting out of the spleen?
Yeah, into the peritoneal space. Yeah, into the abdomen. Right. So it's not in the circulation to sort of.
Blood is not in circulation. It's like a reservoir.
Right, but you got to think about that it's in connection with the capillaries and with the hematologic system, right? I mean, that's why we're talking about the spleen, is that it's connected to that circulatory system. Sure. So it's needed in terms of like, not being hypotensive and maintaining a blood pressure. If you would lose all of that through through some sort of trauma where all that blood is now out, leaking out into the peritoneal cavity or into the abdominal space, then yeah, you're talking about issues with maintaining blood pressure and, right, circulatory flow, etc. It's not a stupid question. No, not at all. Not at all. No. Okay. So again, major functions of the spleen, all the things we talked about, filtering of debris and dead blood cells, and sort of maintaining that reservoir of blood. And the reason that blood is maintained in the spleen is that for some type of emergency reason, if the body needs more blood at the drop of a hat for some other, emergent reason, that we have this backup reservoir of blood. So it's a good thing usually, because you can sort of use the spleen as sort of a backup of blood that we may need in some type of emergent situation. Just a problem when the emergent situation has to do with the spleen. Does that make sense? Okay. Hope that's clear. All right. Now, the last lymphoid organ that I really want to talk about is just really all of the lymph nodes that you find throughout our body. So the way that you can really think about lymph nodes, and first of all, where do we find lymph nodes? Where are they located? I mean, yeah, right, exactly. We have them axially, we have them inguinal, we have actually a lot in the mediastinum, in the chest. You have a lot of cervical chain of lymph nodes, so a lot in your neck and around and behind your ears. Good, very good. So in talking about lymph nodes, you can really think about them as both part of the immune system, so we've already talked about the immune system last semester, and as part of today's conversation, as part of the hematologic system. And the way that they can sort of fit into both of these categories is this. Structurally, so the way that they're actually the way that they're structured, they really are part of the lymph system, which is hematologic system, right? Because they're oftentimes clustered around lymph veins. So you have, not only do you have your circulatory system of, the blood flow, but you also throughout your body have veins of lymph fluid as well that have sort of drained off old debris, old icky stuff from our tissues, and is draining some of that tissue off of the tissues, or some of that fluid off of the tissues. So structurally, lymph nodes are part of the lymph system because they're clustered around lymph veins. They collect interstitial fluid from tissues and transport it as lymph fluid back into the circulation, so back into the blood to then, to be gotten rid of through filtration, through whatever process, that we just talked about, filters out the blood. So structurally, you can think about it as part of the hematologic system, right? Again, I'll say it again. They collect interstitial fluid from tissues in our body and transports it as lymph, fluid, which dumps it back into our bloodstream, which then gets filtered out through our blood filtration processes that we've already talked about. And then functionally, the reason I say that lymph nodes functionally have a role is that they now are part of the sort of immune system because of the way that they can sort of drain off fluid and drain off old debris and, toxic products from the tissue and get it out of the tissue, back into the blood, and circulate it to be filtered. So that being said, has anyone ever had like an ear infection or some type of maybe sinus infection or something like that, and several days later you sort of sense that you've got some like swollen lymph nodes maybe in your neck or mastoid process behind your ear or things like that? This is exactly why. Because if there's some sort of infection in the tissue, let's say in your ear or a sinus infection or something like that, oftentimes that old cellular debris that is sort of building up at an infectious site will then drain through those lymph system, through the lymph veins, and get backed up, so to speak, in these lymph nodes. And the fluid will always go to sort of the closest lymph node, the closest part of the lymph system. So that's why you see, like, when you have a cold or a sinus infection or something like that, oftentimes you'll have some enlarged lymph nodes. It's because of this increased volume, so to speak, of this lymph fluid in the circulation, and because of this sort of infection going on in the tissues. So it also facilitates in maturation of lymphocytes. It does exactly what I just said, transports lymphatic fluid back to the circulation, and cleanses the lymphatic fluid of microorganisms and foreign particles once it gets into the hematologic system and can sort of be filtered out. Okay, so during infection, interestingly, the rate of lymphocytes within these lymph nodes, is so great that the nodes really do start to enlarge and actually become tender. And it has actually not only to do with obviously increased fluid, being drained off of these infectious areas, but also because of the actual increased number of lymphocytes. So increased lymphocytes and increased fluids present is what causes lymph nodes to enlarge during some type of sort of acute infection. And actually, if you look here at the structure again, the cross section of a lymph node, hopefully that sort of enlargement and the swelling of those lymph nodes should become pretty apparent to you. So take a look at this. If you do a cross-section of the lymph node, okay, you have the pulp and you have the sinuses like we talked about. But look at all these afferent lymph vessels that are flowing, so that fluid out of, pulling that fluid out of the tissue that's been infected, pulling it into these lymph vessels and into a lymph node. Okay, you've got 1, 2, 3, 4 afferent lymph vessels dumping into a lymph node. Then they work their way through these sinuses and through this pulp. And now take a look at this. You've got 1 efferent lymph vessel flowing out of that lymph node. Hopefully, it should make pretty good sense to you that if you've got an increased volume of fluid, you've got an increased number of lymphocytes moving through these nodes, you're going to basically get a traffic jam, right? On an immune and sort of structural basis, you're going to get a traffic jam inside these lymph nodes, and that's why you get enlarged lymph nodes in some type of acute infection. Does that make sense? Okay. So again, there's several sort of afferent lymph vessels that bring lymph to a node. There's only one lymph vessel that leaves the node and that sort of pulls that fluid out of that lymph node. So, and the arrows kind of obviously show you the direction of lymph flow. So you're really getting a traffic jam and enlarged lymph nodes. Hint, hint, wink, wink. You may want to know that. All right. So any questions about that? Any questions about sort of the cellular constituents of the blood, the sort of plasma constituent part of the blood, and then any of sort of the lymphoid organs before we move on to actually discussing how our blood is made? Yeah. Yes. Yeah, so the way I would sort of think about it, and the way you can sort of structurally sort of try to kind of organize it in your head, is that in my mind, pathophysiologically speaking, the macrophage system that we just talked about cleans the blood on more of like a micro level in terms of like the lymphocytes and cleaning and filtering out of that old dead blood cells and things like that. But yes, then so yes, it gets circulated through it, and yes, it gets filtered. But ultimately, on a more of a macro picture, it's the liver that does that major, massive cleaning before you send that blood back up into the circulation to get pumped out back by the heart. Does that answer your question? Okay. And do we have another question, or did that kind of answer it? I thought I saw another hand. Okay. All right. So yeah, great question. And again, that's what I mean. That's what I was sort of saying at the beginning of this lecture, is that all of these pieces of the puzzle are now kind of starting to come together for you a little bit, right? The way that the GI sort of liver functions work with the hematologic functions, which work with the immune functions that we talked about last semester. You really are starting to put the pieces of a patient, clinically speaking, really putting the pieces of this puzzle together and seeing how this all really does work together. Great, good question. All right, so that being said, let's talk a little bit about how blood actually gets created, right? So the production of our blood. So hematopoiesis is the general process of blood cell production. And so whenever I say hematopoiesis, I mean specifically, or shall I say generally, all of the blood cells, right? So red blood cells, white blood cells, and platelets. Now, we mentioned this already at the beginning of lecture, really only occurs in the fetus. It only occurs in the liver and the spleen, which is very, very different than the way it occurs anytime sort of post-delivery, and that is only in the bone marrow after birth, okay? Now, I will tell you, and I don't know if you've looked already in the text in terms of this content, I will tell you the text is extremely technical. I mean, you could literally read this content about hematopoiesis for weeks. I mean, it's so technical. Personally, I don't think that it's necessary to be that specific and to understand that level of technicality for the purposes of our job as clinicians taking care of patients. So what I'm going to do today is really going to cover what I think is important and what I think is practical and sort of warrants our understanding in terms of being able to take care of patients, understanding what you're seeing, you know, in terms of lack of blood production or blood production in our patients. So that being said, what you're going to find here is probably not near as technical or as specific as what you're going to find in the text, but what I want you to understand is what I'm going to be going over today, okay? So, that being said, it is a very technical, very complex process that involves, basically biochemical stimulation of a lot of different populations of relatively undifferentiated cells, right, in our body to undergo. So basically, these very undifferentiated, primitive stem cells, basically, that undergo two things. So two stages. One, mitosis, and two, then, maturation and differentiation of those cells. So, that being said, when you think about the first stage of hematopoiesis, think of it as basically the mitotic division or the proliferation, right? The growth, the proliferation of blood cells. And then two, maturation and differentiation is really what I'm talking about when you talk about now taking these stem cells and differentiating them out into mature cells. So differentiating them out into red blood cells, white blood cells, platelets. Okay? Proliferation, so that first stage, that mitotic stage, usually stops after a few of the doubling cycles, right? To actually proliferate and make new blood cells. But differentiation continues almost, continuously. Okay, so proliferation usually stops after a few of the doubling cycles, but maturation and differentiation of those stem cells into red blood cells, white blood cells, platelets, oftentimes will continue. Okay? And this process of mitosis and maturation / differentiation really does continue throughout life. And obviously, hopefully it's obvious, this hematopoietic process increases in response to a lot of different factors that can occur in someone's lifetime. So what are these factors? What are factors that may increase or speed up this two-stage process of creating blood? What are some factors that you think, clinically speaking, our patients would need increased blood or increased blood production? What do you think? What are some times when hematopoiesis is sort of accelerated or sort of asked to do it more quickly or more regularly? What do you think? Yeah. Okay, so hypoxic situations are a huge driver for increased blood production. Very good. So hypoxic, ischemic kind of conditions. I would also lump into that altitude, right? Higher altitude, someone who lives in a higher altitude situation, actually because of that lack of oxygen or in the blood in higher altitudes, actually kicks in the hematopoietic drive and you create more blood. And that kind of is lumped into that hypoxic drive for hematopoiesis. Good. But there's other things too. What else? What else do you think? Infections, disease, good, very good. Yep. Yeah? Yes, pregnancy does as well. Why does pregnancy call on increased hematopoiesis? Do you know? Okay, so because of the oxygen needs of the fetus, good. And I heard someone else say something that was right on. I think the baby's taking your stuff, right? You can really think of, and that's very good. So both of you in combination is a great answer to that. You can honestly think in a pregnant woman that the circulation of the mom is the circulation of the baby. So you need to increase the volume. You need to increase the blood because you're basically sharing it between two. 1 second, Haley, and then I'll come back to you. So in cases of like a singlet, right, when a woman is pregnant with one baby, her hematopoiesis may increase a singlet, right, may increase a little bit, right? You actually may see an increase in blood volume with one. That talking about twins, even more. Talking about triplets, even more. So then you're starting to think about all this excess blood volume circulating in this woman's hematologic system, right? Several years ago, it wasn't Octomom, but it was one of those ladies who had like, 8 fetuses because of, fertility help and things like that. That after she delivered the 8 fetuses, several days after, she went into basically profound congestive heart failure. And that should make sense to you, right? Because now you had 8 babies taking up some of this increase in circulation. Now these 8 babies are gone, and what is this mom left with? Basically 8 times the volume of her blood, 8 times the volume of her circulation. So completely overwhelming her heart and her lungs, right? So that should make perfect sense to you then to sort of illustrate clinically this issue of hematopoiesis in this woman who had basically 8 times the volume. And now she doesn't need 8 times the volume, and she basically overloaded her heart and lungs. So excellent example. Very good. Now you. Yeah. Yes, hemorrhage. Thank you. Probably the most straightforward one. Very good. Hemorrhage, right? Loss of blood volume is obviously going to create a need for hematopoiesis. Good. Yeah.
What about just normal growth and development? Does that
Maybe a little bit. Yeah, maybe a little bit. But it's such a small, slow, steady incline that you really don't see it really kick into hyperdrive. Yeah, yeah. One more back there. Yeah. Yeah. So in the case of like a quintuplet kind of situation, they actually may not have like what they call polycythemia, which is like increased red blood cell count. It's more of the actual whole volume of blood. So it's not just red blood cells, it's the plasma, it's the red blood cells, it's the platelets, it's everything. So it's almost more of just like I said, like a congestive heart failure situation that you've got increased volume, not really increase in any one component. It's all sort of increasing together, and the ratio almost looks the same. Does that answer your question? Yeah. I think that's what they did. I honestly think they fixed her CHF. I mean, she was completely normal and healthy after that. They treated her. It wasn't like she died. But I think it was like therapeutic phlebotomy, like literally draining some liters. Yeah, literally. Yeah. Okay. So good. All of really good factors that would cause the process of hematopoiesis. Okay. So it very much is part of the stem cell system because it's these very primitive stem cells that then differentiate into these important, cells of the blood. And bone marrow is absolutely, anytime after birth, the site of hematopoiesis. So that's why bone marrow is also called myeloid tissue, because it is the site of hematopoiesis. So when talking about our bone marrow, which we have to do when we're talking about hematopoiesis, we actually have 2 different types of bone marrow in our body. We have red bone marrow and yellow bone marrow. So, and this is really specifically talking about, adults. So, I don't really mean adult, but like anytime post-birth, right, non-fetal, is red and yellow bone marrow. Red is your active sites of hematopoiesis, where you're actually actively creating new blood cells. Yellow bone marrow literally looks yellow because it's more of like a reserve fatty tissue in that area of the bone. So it really only creates and sort of helps in the process of hematopoiesis under sort of emergent situations, again, where you need to sort of call on reserves for increased production of blood and sort of increase process of hematopoiesis. So red bone marrow is where hematopoiesis is occurring all the time. Yellow bone marrow is really just sort of a reserved site for increased need of hematopoiesis during sort of emergent situations. But here is a really good list of all the sort of active bone marrow sites in our blood, or in our body, I mean. Okay. So that's kind of the general conversation of total sort of blood formation and the process of blood formation. Now we're going to kind of look specifically at each one of those cellular components of the blood. So specific red blood cell formation is where we're going to start first. So that is the term erythropoiesis. So formation specifically now just of red blood cells. Erythrocytes, so red blood cells, are derived from erythroblasts. So that's sort of the less mature, more primitive version of a red blood cell is an erythroblast. And the only reason, really, that erythrocytes become erythrocytes and mature from an erythroblast to an erythrocyte, it's stimulated by, again, a hormone, erythropoietin. Okay? Erythropoietin causes an increase in red blood cell production in the bone marrow, and then also causes the bone marrow to release it out into the circulation in times of need, in times of need for increased red blood cell presence. Now, Erythropoietin is the hormone that stimulates erythrocyte production. Where does erythropoietin get secreted from? The kidneys. Perfect. Very good. Erythropoietin is secreted by the kidneys. It's a hormone, so then it travels, it gets secreted into the blood from the kidneys, travels to the bone, and stimulates the bone marrow to create more red blood cells. Right. So again, I hope it's not that crazy of an idea for you to think about that when we talk about the renal system in weeks ahead, that someone with chronic, kidney disease or renal failure might just also be what? Anemic. I wonder why. Erythropoietin, right? Again, starting to put the pieces together. Good. So it's not unheard of at all for chronic kidney failure patients to be profoundly anemic because of their lack of secretion of erythropoietin. Very good. All right. So why? When does the kidney, under normal, healthy, functioning kidneys, when does the kidney know to create erythropoietin? Yeah. It sends some hormones, right, exactly. So not only would blood flow, so lack of blood flow to the kidneys be a big sort of drive of secreting that erythropoietin, and what else? So very, very along that same lines, it goes with that. Hypoxia, lack of oxygen, right? When the kidneys sense lack of blood flow / lack of oxygen, it's going to actually secrete that erythropoietin to the bone marrow to create more red blood cells. Very good. So hypoxia, to be clear, hypoxia is the trigger for erythropoietin secretion. So in response to any type of tissue hypoxia, it causes basically a compensatory increase in the erythrocyte production, if oxygen content of the blood is low. And this could be because of anemia. This could be because of higher altitudes, living at a higher altitude location. It could be because of pulmonary disease. So if you've got like COPD and you're breathing in, able to sort of get increased oxygen into your blood and you're a little hypoxic because of some sort of pulmonary disease, your erythropoietin will kick in and try to create more red blood cells. Very good. So anytime there's not enough oxygen presence, erythropoietin is going to kick into gear and create more red blood cells. So here's just kind of a picture of exactly what I just said. So here's some more reasons for why you might see regulation of erythropoietin, or shall I say increase in erythropoietin. Decreased red blood cells, so if someone's anemic, you're going to see erythropoietin kick into gear. If you see a decreased hemoglobin synthesis, so you've got decreased hemoglobin on those red blood cells. You have a decreased blood flow, as Blake mentioned. Hemorrhage, loss of blood in general. And the one thing we didn't necessarily say, but I'll say it here because it's on the screen, is an increased oxygen consumption by the tissues can also increase and sort of stimulate erythropoietin and erythropoiesis. What would be some examples of that? What would be some exercise and seizures? Very good. Very, very good. When the oxygen consumption is so high and being used up in the muscles, your body needs more red blood cells to go out there and compensate, right? Very good. Very, very good. And this is a hormonal function, right? Erythropoietin is a hormone. It's part of the endocrine system. So here's a kind of a visual. So let's say there's decreased red blood cells here in the circulation. That triggers the kidney to say, we need some more erythropoietin to send out to the bone marrow. So it secretes erythropoietin to the bone marrow. Bone marrow makes more blood cells, plops them in the circulation. You've got this good normal number of erythrocytes now in the circulation, and it goes back as a negative feedback loop and tells the kidneys, all right, that's enough. Thank you. We've got enough red blood cells. You can stop creating more erythropoietin, right? So it's a negative feedback loop. As you create what the body needs, it goes back and turns off increased secretion from the kidney. And we'll talk all about negative feedback loops when we talk about hormones and endocrine function in a few weeks. But this is the great picture of that role of erythropoietin in red blood cell formation. Okay. We also need to talk just a little bit about actual hemoglobin synthesis too, because remember I said it's not just enough to have red blood cells present. right? You need hemoglobin on those red blood cells. So hemoglobin is obviously that oxygen-carrying protein of the red blood cell. It enables, the reason that we care so much about hemoglobin is that it enables the blood to transport 100 times more oxygen than it could be transported just, dissolved in the plasma alone. So there is some oxygen that's dissolved in the plasma circulating in our blood, but it is so much more effective to have oxygen being carried on the hemoglobin. It increases the amount of oxygen in the blood up to 100 times. And that's because, this next step, a single red blood cell contains as many as 300 hemoglobin molecules on it. Anyone know how many oxygen molecules are on each hemoglobin? 4. So you do the math, right? About how much that increases the oxygen carrying capacity of the blood if you've got a good number of hemoglobin molecules present on each one of those red blood cells. So that's why we care. That's why we care about hemoglobin synthesis in our patients. Okay, very good. All right, any questions about red blood cells and the functions of red blood cells before we talk a little bit about development of white blood cells? Okay, so development of leukocytes. Again, leukocytes arise from the stem cells, right? Just like we talked about when we talked about the overall sort of general blood formation. Leukocytes arise from stem cells just like red blood cells do in the bone marrow. Granulocytes mature then also in the bone marrow. And agranulocytes and monocytes are released into the bloodstream before they fully mature, right? So granulocytes mature in the bone marrow, but agranulocytes and monocytes are released into the bloodstream and then go to some of those lymphoid organs. Thank you. Oh my God, I have a brain fart. Those lymphoid organs to go and do their maturation. Okay? Good. Also, when discussing this development of leukocytes, other things sort of play into leukocyte formation. So growth factors and colony-stimulating factors encourage the production of this maturation of leukocytes. And obviously, production of leukocytes are going to increase during times of infection, increased presence of steroids. Why? Why would steroids actually increase or cause the increase, so to speak, of leukocytes? Why would that be? Anyone know? What do steroids really do? What are they sort of known to do or known to cause? Mm-hmm. Right, so steroids are anti-inflammatories. They're also a little bit of like an immune suppressant, right? They actually sort of suppress the immune functions a little bit. So if the body senses that there is immune suppression, what's it going to try to do? Kick up some white blood cells, right? And then the next things that I have here on the screen, like increased heart rate, pain, nausea, and vomiting as a cause of development of leukocytes, I'm going to kind of amend that and really say that oftentimes these obviously are clinical manifestations. They're sort of symptoms of some of these things like infection and things like that. And so that's why you oftentimes will see leukocytosis, so increased white blood cell count, when you see things like increased heart rate, pain, nausea, and vomiting, because they're oftentimes going hand in hand with some type of infection, which will then increase the white blood cells. So I just wanted to mention that. It's kind of a misnomer the way I have it up there on the screen. Okay. There's usually something else going on if you're having an increased heart rate or pain or nausea and vomiting, and that's why you see the white blood cells. Okay. Much briefer discussion of development of leukocytes than of red blood cells. Any questions about that? Okay. Last but not least, development of platelets. So platelets develop under a process called endomitosis, and we've kind of already discussed this today already. So platelets develop from cells called megakaryocytes. So megakaryocytes are your more primitive version of a platelet. And they're developed from megakaryocytes by a process called endomitosis. So again, the megakaryocyte undergoes that normal nuclear phase of cell division, where the genetic material doubles up and gets inside of, on two sides of the cell. But now the cell fails to undergo cytokinesis. It fails to sort of create and pull apart and make two new good normal cells. So what happens is that the megakaryocyte expands due to the doubling of that DNA inside of it, but instead of sort of splitting into good two cells, it sort of breaks up into these fragments. And that's what platelets are. They are these cellular fragments that have occurred from this process of endomitosis. So, platelet levels are maintained, imagine that, by another hormone, this time called thrombopoietin. Hopefully that should make sense to you. Thrombus, clotting, right? Thrombopoietin. Platelets circulate, again, here we go, for 10 days before losing their ability to carry out some of their biochemical reactions. So also, very short lifespan, even shorter than the red blood cells that we talked about. And these old, and I say sort of senescent, right? Old platelets then get sequestered in the spleen. They get sort of held up and sort of reserved in the spleen for what? For what to happen? The MPS to destroy it, right? To filter out and destroy those old, nasty, non-functional cells we talked about already, right? So again, we mention it again, like we talked about. Okay, so what's going to cause thrombopoiesis? What is going to cause hemostasis? And basically hemostasis means the arrest of bleeding, right? The clotting, which is what platelets do. What kinds of things are going to cause hemostasis? And by the way, is hemostasis a good thing? Is blood clotting a good thing? Absolutely. We think of it clinically, right? We think of it as bad. Oh, a blood clot, that's not good. But obviously, under most circumstances, we need hemostasis. I really don't want to bleed out and hemorrhage from a paper cut. Okay? Doesn't seem very efficient to me to worry about having to bleed out from a paper cut, and that is because of hemostasis, right? So it's a good thing. So what are the requirements for hemostasis? So obviously platelets, hopefully that's pretty self-explanatory, clotting factors, and endothelial cells of the vasculature. Okay, now hold on a second. I said hemostasis occurs with sort of the requirements of platelets and clotting factors. Why would the third one, endothelial cells of the vasculature, which basically I mean like the endothelium, right, the lining of the vessels, why is that a requirement for clotting? Why is that a requirement for hemostasis?
Like chemical signals?
Yes, to start the cascade, basically. So clotting occurs because of endothelial cell or vessel injury that causes the clotting cascade. So to be clear, endothelial injury, so the injury of the lining of the vessels, has to happen for hemostasis to occur. So I put it third on the list, but really almost I should put it number 1 on the requirements for hemostasis. I'll say it again. Clotting occurs, hemostasis occurs, because of endothelial cell damage or endothelial cell injury. Because that is the instigator of the signals that get sent out to actually then cause the clotting cascade to occur, to cause hemostasis. So that damage to the vessel could be my paper cut, right? It could be something worse, like a huge trauma. It could be atherosclerosis, right? We talked about last semester when we talked about heart disease, how atherosclerotic plaques can damage the inside of the vessel and then cause a clotting cascade, right? So it doesn't matter what the injury is to the vessel. Whatever it is, it causes now the same clotting cascade. It causes the same process of hemostasis, which looks like this. Now, I apologize. This is the writing over here is extremely small, but the pictures are pretty good. So let me kind of talk through each one of these stages of hemostasis with you and sort of make this a little bit clearer for you. So again, first off, blood clot is just basically a meshwork of protein strands, right, that stabilizes the platelet plug that sort of originated in that area of vessel injury and traps the other cells, such as red blood cells and phagocytes and actually the microorganism that might be at that site, and really creates that platelet, creates that hemostasis. So again, step 1 here, subendothelial exposure, right? The lining of the vessel wall has to erode away. It has to have some type of injury or damage to it where it exposes sort of the subendothelial layer. This is, again, ding, 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 the injury, right? This is what that sends out that signal for everything, all those important platelets and all those other important blood cells to come to the area to migrate to that area. So 1, exposure, injury of the vessel wall. That's what 1 is, injury. 2, what happens? Well, first thing that happens is that the platelets kind of start to adhese together. So the second stage is called adhesion, right? And adhesion is initiated by the loss of those endothelial cells of that vessel wall. So they start to get sticky. They start to stick together, okay? And they kind of start to settle in now during this first part of adhesion. They kind of start to settle into that area of damage where they start to sort of aggregate in that area. But true aggregation doesn't happen until a little bit later, but they start to. But the biggest thing I want you to remember about 2 is that they start to adhese. The platelets start to get sticky. 3, activation. After the platelets start to get sticky and after they adhere, they undergo sort of an activation process that leads to sort of the activation of a lot of other processes and a lot of other cells coming to that site. So here in this picture, you see collagen starting to form. You see red blood cells coming to that site. So in stage 3, I want you to think of the activation of bringing other cells, other important cells in the blood, to that site of endothelial damage, in addition to the sticky platelets that already showed up. Now, step 4 is aggregation. This is where you start to see the aggregation of all of those cells in that site. It's also now where you start to see the fibrin mesh starting to form. Why is the formation of the fibrin mesh so important during this phase of aggregation? Why is that so important that we have a fibrin mesh appropriately and sort of effectively form in that area? What's the process? What's the point of that? Yeah. Yeah, right. It keeps the bad stuff there, but it also keeps the good stuff there to fight that bad stuff, right? So it keeps all that important stuff in one site so that it's not all floating around and becoming diluted and not sticking to that site of damage to do its work. Very good. So that's what's happening in aggregation. Then in 5, platelet plug formation, you really start to see not only the platelets, but also the red blood cells really get enmeshed in that fibrin, right? In that basically that cage, right? That little net that's over that site. And you start to see a blood clot form, a platelet plug form. And again, that's why the fibrin sort of mesh is so important there, because it holds all those red blood cells and all those platelets there. And we know that when blood sits, specifically when platelets sit, what happens? Clot formation. Right, exactly. And in this case, we want it to form. We want it to be a plug in that damaged endothelial wall. And that's what you see here in 5. Now, last thing is clot retraction and clot dissolution. So I've already said how good it is, right, that we normally, under normal circumstances, have clot formation. But do we want that clot sitting there forever? No, right? So what has to happen? Eventually, the clot needs to retract and dissolve and go away. We don't need that clot sitting there forever. So what happens here under 6, clot retraction and clot dissolution, is you'll start to see the sides of the endothelium, or the sides of the vessel wall, start to kind of get, from this clot formation here, start to kind of get pulled together so that the edges kind of start to meet each other. So not only do we have the clot starting to be dissolved and starting to retract, but we're also seeing sort of healing and closure of that endothelial wall. Anyone know what substance actually causes the clot to start to dissolve and sort of break up? I heard it. Say it louder. Plasmin and plasminogen. Yeah, very good. Okay. So, clot dissolution is basically regulated by plasminogen. We're going to come back to this in just a second. Any questions about those processes? Again, the wording off to the side is extremely technical. It's extremely small. I wanted my explanation to sort of be what you understand from each one of those pictures. Okay, so basically, here's another really good picture of the blood clotting mechanism. Basically, this complex sort of process can really be distilled and sort of whittled down into 3 basic steps. 1, you've got the release of clotting factors from an injured tissue. So here's the damaged tissue or the damaged endothelial of the vessel, right? 2, you have a series of chemical reactions that eventually result in formation of a clot, so formation of the thrombus or the thrombin, right? And 3, you get formation of fibrin and the trapping of all of those blood cells to form that clot in that area, right here. Okay. Well, 1, release of the clotting factors from the injured tissue, so that initial signal that the injured tissue sends out. And then 2 is the chemical reaction that sort of eventually results in the formation of thrombin or of the clot. And 3 is the formation of that fibrin mesh trapping all those blood cells at that site. Well, it's basically what I just said on the previous screen, just kind of distilled down into 3 steps instead of 6 or 5 or whatever it was. Okay, so once you've got those 3 blood clotting mechanisms, checked off your list, you then also do have to talk a little bit about what we just talked about in terms of clot dissolution. So this is just kind of reiteration of what I've already said. It's kind of some slides here to be a little bit more specific for you regarding clot dissolution. So, clot retraction. After a clot formation is sort of formed around that injured vessel and it stops the bleeding, it needs to dissolve. And after a clot, and after platelets sort of contract and pull and that fibrin threads close those vessel walls together a little bit more, you start to have the clot not needed anymore. And you start to have the clot broken down by plasminogen. And let's talk about that here on the next slide here. Okay, plasminogen is converted to plasmin, and plasmin is an enzyme that actually just starts to break away and dissolve the clot by degrading that fibrin mesh and degrading that sort of little cage or that little basket that was sort of created there. It sort of breaks that down. So it truly is a balance between the amounts of thrombin, which create a clot, and plasmin, which break down a clot. And again, I want that to be clear to you. Thrombin is needed in the formation of a clot. Plasmin is important in the breakdown and the dissolution of a clot. And it's this balance between these two in the circulation that maintain our and our patients' normal coagulation and lysis. Okay. And this is just a really good picture of what I just said. You have plasminogen, which is activated by something called TPA, or tissue plasminogen activator, which activates the plasmin, which starts to actually degrade that fibrin clot. Okay? And I will tell you that the fibrinolytic system right here is one of the objectives. So just make sure you understand this picture of what I just said. Plasminogen is activated by TPA, which changes that into plasmin, which is what actually works on the fibrin clot to break it up. Okay? And that is it for today. The last 2 slides are just some of those considerations regarding the pediatric end of the age spectrum and the geriatric end of the age spectrum in terms of differences that you see in those populations within the hematologic system. On Thursday, we'll talk more about alterations of this system. This whole process, I'll do it on the screen so you can see it in the recording. Plasminogen is activated by TPA, tissue plasminogen activator, to create plasmin. Plasmin now is what actually directly impacts the fibrin clot and breaks it up. Okay? Sure.